a researcher with USDA ARS at the Brooksville station for many years and was a collaborator both with myself and other colleagues at the University of Florida IFAS. And David has been an important component of some of the Angus work that we've done at ONA, and a frequent discussion, included in frequent discussions about the coloration of the ONA White Angus. And David's an expert in this area as a geneticist and animal breeder. And so I asked him if he would join us today and help explain the genetics behind hair coat color in beef cattle and help us along with that topic. So thank you, David, for making the trip, and pleasure having you.
Thank you, John. Let me dispute the term expert. Okay, I sort of prefer the term continual student, okay, because expert implies something that I'm really not. So a lot of you probably already know a lot about color inheritance in cattle, and I don't want to insult you at all, but I'm going to review things very basically from the very beginning. We teach this to our undergraduates at Texas A&M, and I'm sure you teach something similar at the University of Florida. So I think color, if you go back and look at the history of cattle breeding, it was one of the original things that the first guys, the Bates, the people like that in England who were selecting cattle and trying to improve them, that was one of the first things they started doing. You know, the Hereford and the Shorthorn registries were the very first ones, and I think because it's a big, big phenotype that we can see and change easily, that was one of the first criterion that people used to improve livestock with. Absolutely, it has economic value in the United States today. Nobody would dispute that at all. You can see that by the number of breeds that were not originally black that now have black animals in them, right? I mean, you can't deny that when they go to the sale ring, they bring more money with a black hide. There's other aspects of economic value. For example, think of Brendel females, okay, and what kind of value they command in the sale ring versus Brendel steers, okay? One group would be very costly and dear to producers, and the other group would be almost a byproduct, right? So as Joe just mentioned, there is adaptive value associated with coat color. And the most obvious one is the ability to radiate heat, or they absorb heat, which is why black cattle are in the water or they're in the shade early in the morning in Florida all through the summer, right? There also is some brand recognition. Almost everybody in the United States, we get a lot of students from inner city Houston, and almost all of those can recognize the black and white pattern of a Holstein cow, right? And many of them can recognize a Hereford. How universally recognized is a Hereford in our world today? So it is a system of brand recognition. It facilitates, okay, I'm employed in this and I have a career in this, but I will tell you that the study of inheritance is quite complex, and these are nice big phenotypes for us to look at. In other words, we get large discrete changes associated with cattle color, and that helps us understand inheritance in mammals. So like I said, a really big phenotype. A phenotype is something we observe on an animal. You could go weigh an animal at a year of age, and we like to call that a phenotype. Okay, well, its color is absolutely something big that we can see and classify. So a black versus a red. A lot of the work stuff that is known is based upon my friend and colleague who was at the University of Florida for many years, Dr. Tim Olson. He did his PhD dissertation at the University of Wisconsin on color inheritance in cattle. He also published that in a book in the late 90s, and I'm sure that if you had a desire to see that, I could find that reference for you. I'll also, my mentor at Texas A&M is Dr. Jim Sanders, and he's absolutely obsessed and has more passion for cattle than any person I know in the whole world. But he has taught me so much, and he has thought so much about the inheritance and observed inheritance of color and marking in cattle as much as anyone I know. So a lot of what I'll report to you, I will use designations that are sort of unofficial, okay? in many cases, we are moving into the molecular era, and we can tell you what gene is responsible, and we can go find that in the genome and name it and categorize it molecularly. But in many cases, we're just looking simply at quantitative inheritance. And without being official or having official designations, that's what we'll kind of talk about. So I'm going to talk about 5 areas with regards to color inheritance in cattle: a base color, and the genes that we see or the genes that have been identified as responsible for these areas. I'm going to talk about gray color dilution in cattle. We'll talk about brindling, and finally also about spotting. So there's 2 types of genetic action, okay? And we teach this in courses, but it's helpful for us to understand this type of inheritance. One is qualitative. In other words, the things we observe fall into giant categories like this is a red animal versus this is a black animal. Or this animal has horns, this animal is polled. You see what I mean by qualitative? It is a big, almost yes-no type trait. Now, most of the things that are important to you as cattle producers are quantitative type traits. In other words, there's a whole bunch of genes responsible for those, and they vary continuously. So in other words, for example, birth weight. We can arbitrarily put that into categories, right? But in reality, birth weight falls on a number line, right? From about 50 pounds to about 150 pounds, and you can infinitely divide that number line, right? Forever. You could get a birth weight of 90.12697. You could really get some fine shades of differences for a continuous variation trait. And just as I said earlier, I mean, those are typically controlled by a bunch of genes, whereas qualitative traits are controlled by just a few. So they're easier to study, okay? I always tell my graduate students, all the easy stuff has been done, okay? You get your PhD, you get something, you have to do something more than we did, right? Because all the easy work we did, you know? So good luck to you on that, but it gets more complicated as we go. All right, here's some terminology. Again, a lot of you know this, and I don't aim to insult you, but let's review it. It helps us be on a common ground. But a genome, in every cell in your body, and every cell in every one of your cows' bodies, they have 2 long strings of DNA, okay? And they pair with each other, they correspond to each other, and we call that the genome. They're made up of nucleotides, and we don't need to know the specifics on that, but you need to know that they each have 2 strands. One strand comes from paternal side of their pedigree, and the other strand comes from the maternal side of the pedigree. So a locus, you can kind of remember that term by using the first 3 letters. That is a place. And I slip and use that word a lot, but a locus is just some place in the genome, okay? It may be a gene, it may just be something that we see, a marker, rather than a gene. And then I like to define a gene as, well, that is a region of DNA that actually produces a protein that results in a product, okay? So get some of those genetic terms out of the way. Allele. That sounds very scientific, right? But allele, there are multiple forms of genes, okay? And each gene then is made up of 2 of those because we have 2 long strands of DNA. So we have 2 versions of each gene. Sometimes they're the same, okay? But each version we designate to be an allele. And then we call a pair of those alleles a genotype, okay? If the alleles are identical in a genotype, we call that homozygous condition, or we call the individual a homozygote, like homo meaning same. And if we have an individual that has different alleles at this particular gene, like in the bottom one there, I'm using an upper Roman case E to indicate one allele, a lowercase Roman E to indicate another allele. Well, that would be what we would call a heterozygous individual or a heterozygote. So, and the key part of the word being hetero, different, okay? So pigmentation is really cool how it happens in mammals, okay? It happens very early in embryogeny. That is right after conception, and the embryo starts to form, and as it masses cells, once it reaches a certain point, That's when this melanin starts happening, okay? The embryo is oriented in utero, okay? And its orientation, it's really neat because the DNA uses its orientation to determine what part's going to be dorsal and what part's going to be ventral, okay? In other words, where are legs going to form? Where is the head going to form? It's all spatially determined by how the embryo is situated. And that's really interesting. Pigmentation happens that way also. Okay, so once melanin starts happening, and once it starts traveling through the coat of the new embryo, okay, it starts from the top and goes over the sides and down. Now think about a Hereford pattern, okay? Does it seem to make sense to you now? Think about red pigment starting at the top and moving across the sides, okay? And something inhibiting it from going all the way to the feet and from going all the way to the head, okay? That's how pigmentation works, and it happens very early in pregnancy, okay? Okay, so the first one we'll talk about is base color. And we term this particular gene the extension gene. That is the extension of black pigment throughout the body. Okay, it is characterized by two alleles, and I'm using this uppercase and lowercase e to designate these alleles. This is not official scientific designation, but we have dominant gene action. In other words, one allele completely masks the effect of the other one when it's present. So therefore, if I have an animal that has a genotype at this locus of two big E's, it is black. If I have an animal that has one big E, it is also black, okay? So that's dominant gene action. There's a third allele at this one with this, got that E with that little plus up at the top, okay? We call that the wild type allele. And that's a difficult concept to convey to new genetics students, but the wild type allele is the allele that's present out in the population in the most numbers, okay? And it's sort of arbitrary now how we designate the wild type allele. But I will tell you that all of your indicus type animals, they are predominantly this wild type allele. And then other things start interacting rather than red versus black color. So yeah, here's your genotypes in the top row. If an animal has any one of those three genotypes, and remember I'm designating alleles with those letters, then what it looks like when we see it is completely black, okay? So that's easy. If it's little e, those are your red animals, okay? We get, like I said, our dynamics of our department are changing a lot, but a lot of our students at A&M now come from the Houston and Dallas Metroplex areas in San Antonio, and they don't know which end of a cow gets up first, okay? But, so they think red means the color our football team wears on Saturday afternoon, you know, so we have to explain to them what red is, but you know, reddish brown, that type of deal. Now these wild type, there's some other things that take over, and we'll try to hit those a little bit. We do know the gene that is responsible for this. It's called melanocortin-1 receptor, okay? MC1R, and it's located on chromosome 18. Whenever you see the abbreviation BTA, that means Bos taurus. That is a species designation for chromosomes, and then we number chromosomes from largest to smallest. So the largest chromosome is chromosome 1, the smallest is chromosome 29, and then we designate the sex chromosomes separately. So like an X and a Y chromosome. But we know where this gene is. This is homozygous little e. Red coat color is recessive, okay? This bull is from Crooked Lake Ranch. My friend David McCullers gave me this picture, okay? So just down the road, a beautiful Victor Domino line, Hereford bull. Okay, so the Angus population in the United States is mostly big E alleles. There's like maybe a frequency of the little e allele of about 5% in the entire U.S. population. So that's why you will occasionally pair animals that will have a red offspring, okay? And that happens at very low frequencies in the Angus breed. These are from our Angus herd at Brooksville. and we would see probably one red. Once we would identify a cow as she had a red allele, in other words, she's black, but she's hiding that red allele, then we sometimes would pick up that she would have a calf that would be red. Okay, so what was simple inheritance about that? And that's very simple inheritance. It turns out that even though we consider this to be a qualitative trait, okay, a really big phenotype that we can learn and study, it's a little bit more complex than that. So my colleague, my student who's now at North Dakota State University, this was her research. We had a research population that we specifically designed as a cross between Angus and Allele, and therefore every animal in the population had this genotype, big E and then the wild type allele. So every animal in the population should be black, right? That's what we said. So what she found was they were not black, and there was a modifier that was changing the outward expression of that color, and she located that to be on chromosome 6 in cattle, very close to a place that we know is responsible for white spotting. Okay, and that place is called the KIT locus. Okay, so, and like I said, what we thought was simple inheritance really is not. Okay, so obviously this was black. By the way, this is F2. You know what F1 is, right? The first generation cross. We used Allele bulls on Angus females. Okay, so we made F1s. Then we made a giant mapping project by mating F1 bulls to F1 females. And those were F2s. And this female is an F2 Allele Angus. Okay, so obviously her genotype is E. You know, she is a black individual. But this individual has the same genotype as the cow in the previous slide. Okay, well, you see this is some variation in black coat color that we did not plan on seeing. Okay, this is really light colored. And then this steer is also the same genotype. So our joke is to drive through the pasture now and look at these and go, oh, you see that black steer over there? Or you see that black cow over there? And it looks just like this, okay? I always key on the insides of the ears, and that's not always accurate. My colleague and mentor always keys on the udder or the scrotum area, okay? He looks more at skin area and that to see what he thinks genotype is, and sometimes we just have to say, we're going to go get blood and genotype this individual and find out what they really are. But black is not always black as you know it. Now this is Angus and Allele cross, and I recognize that's not a real common cross in the United States, but this is where we first found this. He did a nice publication in Genetic Selection and Evolution for Dr. Hanna. Okay, so gray is an important color in Florida and in Texas. And in general, the animals that are gray are the genotype on the right, E+, although often we'll see small e at the base color. In other words, if you've got black, it will sort of override the gray locus, okay? So let's call this two alleles, big G and little g, and so there's dominance action in this one too, and the recessive genotype is the one that gives gray coat color. Okay, so that's how it goes. But you need to have a non-black background in these individuals, okay? So this is a bull that we raised at Brooksville, a classic, gray that you run into. You get a lot of variation in gray as well. By the way, this was the son of a bull that was loaned to me by Ralph and Sean Sexton at Treasure Hammond Ranch. So we had tons of daughters, both straight bred and crossbred out of this bull in our projects there. The amount of black that you see congregating at the ends of the animal, I think, that has something to do with genotypes at other locations, but don't you see that vary with the time of the year in certain animals? We really do, of course. Again, this was at Brooksville, probably 10 years ago. It's the same gene that you would see in Allele. Okay, how do you know this is an Allele? By the hateful look in his eye, right? No, they have a little bit different look to them, okay? But Allele have the same gray, and yet I think Joe said it very correctly. Brahman, it was sort of formed as a composite in the United States based upon Allele individuals, okay? We think that was one of the largest contributing breeds to that breed. Same gray gene present in that one. This is an F4 Nelore Angus. Okay, so you remember I said we'd use Nelore bulls and Angus females and made F1s. We use those F1s. If you made an F1 to an F1, you get an F2. Keep following that logic, and this is F4, okay? And this is this year, you know, off my camera phone. We're getting a lot of color segregation, but we also are picking up gray animals once again because we're making so many back crosses through that thing. By the way, we will breed our first F4 females to F4 bulls to produce F5 next year. That's a genetic resource that I'm very grateful to, that nobody else except the mouse people and plant geneticists have access to in the world. Okay, so I had a student, he's from Louisiana Tech, and I took him provisionally. He had to make good grades before I would accept him as a student, but he turned out to be someone pretty competent and pretty capable. And he used our F2 mapping population, and he found the gene or a region in the bovine genome responsible for gray coat color on chromosome 6. In the same area that Dr. Hanna found that modification of black phenotype, this is also near the spotting locus, the KIT. Okay, so this is really exciting stuff for us, and this will be the first report of the mapping of the region for gray coat color in cattle, and I'm quite proud of him for doing this. Okay, so let's talk about dilution for a little bit. There are at least two types of different action of dilution. One is associated with Charolais cattle, okay, and one is associated a little bit differently for Simmental cattle. And then I don't know if you're familiar with the African Sanga breed called Thule, but we also see a lot of dilution in that particular breed as well. So let's just say, again, no official designation, but uppercase Roman D is a dominant, is one allele, and lowercase d is a recessive allele. But this time you get distinct phenotypes, in other words, distinct colors for each one of those three genotypes. So little d just means there's no dilution going on, okay? And you just have a normal color. You can think of big D, little d, remember heterozygote from my previous slide? That one would be sort of an intermediate phenotype, like on black background, that would be smoky colored calves. Okay, like you see Angus Charolais crosses, we call them smoky. Or if it's Hereford Charolais or something like that, we call those straw or yellow in color. And then big D would be the total Charolais white, like you see. Simmental, I'm not going to go through it. It's kind of the opposite action, but similar, okay? So these are some Charolais and Angus calves in Nancy, Kentucky at a friend of mine's place. But he did a lot of crossing with those. This is the classic Charolais looking. The skin on these is a little darker than I typically see in a Charolais. You generally see more of a flesh-colored nose and skin color. So yeah, here's just a description. If you've got a base color genotype that would result in a black coat, then imposing big D, little d on that makes some sort of smoky colored straw. The gene for this one is known as well, and a German group found this one, and it's on chromosome 5. So they know, they do a lot of work with Charolais cattle, and they know where that gene is. And I'm sorry, I know the abbreviation for the gene, but I don't remember the name. So if you need it, let me know and I'll send it to you. But skin color, very related to base coat color, okay, like the black versus red. Okay, Brendel. Brendel primarily requires a red background, although I will promise you I've seen what I call black Brendel cattle my whole life, okay? And very common when you start looking at Brahman crossed with red breeds, either Red Angus or with Hereford, okay? And Brendel, like we said earlier, it adds such value, at least in Texas, and I know it does here too. It adds value to females if they're Brendel in color, because you know that they're going to express some hybrid vigor for reproduction, right? And it decreases value of steers that you've put in the sale barn to go to the high plains, because those guys go, oh, those are cattle from South Texas, they're going to be sick. When we get them up here, we don't really want those, so they bid them down a little bit. Buyers are among the smartest people in the world, you know? So yeah, there's, again, we don't know a lot about this. We watch the action. There has been a group that has mapped this they mapped this in Jersey cattle, crosses of Jersey cattle. onto a locus they call agouti, which is also one that is for mouse color too. That's a common mouse color on chromosome 13. So it adds so much value to cattle. If we could control this one, in other words, if we could make it sex specific, we could make a mint, couldn't we? Making it be expressed in females and not expressed in males. Boy, that would add some, that would be a research accomplishment, I think. So what's required then particularly is to have one of those two base color genotypes and then have, I've got two alleles, and as long as you have one big B, we'll say we see Brendel. And that's the kind of action that we see with this particular locus. Now, a second one we think is responsible for the black nose, feet, and tail, and also the tips of the ears. And this is all gigantic speculation, but speculation is fun, right? So again, once you have the big B, okay, then if we have a large A, we get some Brendel, but if you get the homozygous small a, that's when we get a different phenotype. You get those individuals that are born, for some fraction of those are born red and turn dark, and it's kind of a restriction of black pigment to the points. If you're familiar with using the term points on horses, then it's the same kind of thing. Nose, feet, tail. You'll often see shiny black noses on those individuals and the black tipped ears, okay? So this is just Brendel calf on one of our projects at Brooksville. Very light colored, obviously. That's the same calf, but brindling is just black pigmentation in non-black background, right? So you can see it in a variety of different background colors. This cow, look how brindled she is, okay? You see it in pit bulls too. You ever seen a pit bull that's brindled? Beautiful dogs. I mean, just beautiful dogs. Now, I put this in and it's not going to show up to you, and I hate when a speaker says, you can't really see this, but this calf is really black brindled, in my opinion, okay? So it definitely looks black coat color, but there's some brindling in there that you wouldn't expect to see. So sometimes in those Brahman crosses and Nellore crosses, we will see that sort of brindling in black animals as well. He's as big as his mother right here. He needs to be weaned. Okay, so this picture is in here so you can see black rimmed ears. Do you see that? Okay, and her nose is a very shiny black nose as opposed to just a dull black. Or you also, if you could see her feet, she has black feet. A lot of times you'll see on the front of their legs, black will kind of go up their legs a little bit too. That I think is the same gene, the same action. And then her switch is definitely black, although we've watched for years, we've watched Brahman with white switches, and observing that sort of thing has always been sort of interesting to me because we should be figuring that out, right? We haven't done it yet. Okay, another black rimmed ears, obviously not as extreme as the previous one, but she also has a shiny black nose and a black switch, and she's either gestating at this point or dry and not working for a living, but this was at Brooksville as well. With your UF brand on the side. So this was one I always observed, is those with black tipped ears like this cow right here, they're the ones that tend to do the color changing as the year progresses, okay? And I don't just see it in bulls, and I would say you probably don't either, but definitely bulls would darken up on the ends, right? And I have cows that would do it too as well. And I have been places where I saw Brahman cows that were essentially black. One of them was in Rockhampton at the Belmont station, which they closed and sent all of the Brahman, they sold all the Brahman cows, and then Brooksville did exactly the same thing Rockhampton did in Australia. So all the Brahman research in the world went just like that. So, but yeah, these darken up and they change across the year, and I mean, it'll surprise you. You just go, wow, you look familiar, but you're just not the right color that I remembered you the last time I saw you if you don't have daily interaction with your cattle. And look at her feet. I mean, that's really black points, right? Super Brendel back there behind her too. You see that cow in the back? With no black nose. She has just a normal flesh-colored nose, right? And no black in her ears. Okay, so let's talk spotting. This is known, this is pretty well documented on chromosome 6. This is called KIT locus, and I'm sorry, that's Dr. Hanson can probably explain that name, but I can't remember. It's some receptor, a kinase something receptor. So this is a locus where there's lots of different alleles present. Okay, so I'm going to use these letters to indicate these types of alleles. And what we have at this locus, sometimes we have complete dominance. So in other words, one allele masks the effect of the other. Sometimes we don't. Sometimes we have sort of codominance. That is, each allele contributes something to the phenotype. Okay? So, for example, a non-white spot allele would be uppercase S. That would be what you'd see in Angus mostly, although you and I both know we see some white on udders in Angus quite a bit, right? So that's fairly common. The Hereford pattern, I mean, that is so clear. Anybody who, you know, that has to be one of the first things genetically that we started seeing and knowing something was happening, is to be able to look at the intermediate phenotype between solid Angus and a Hereford spotted type animal. You know, you get a white face, right? We have a big white face project right now in pigmentation. We want to learn, and what I want to be able to do, and this group is also part of that research as well, but I want to be able to get a DNA sample from a Hereford bull and be able to tell you with high probability that he will sire calves with pigmented eyes. And that pigmentation around their eyes will be sort of a protection against solar-induced cancer. So that's one of our big projects we work on at this point too. So paint side, you've seen that in Longhorns, right? When we teach our students, we put a little superscript P on it, and that's for Pinzgauer. If you're familiar with Pinzgauer cattle, or if you go look at them, they've got that paint side. There are a lot of those breeds in the world that have that paint side kind of look to them. Speckling, the G there, that superscript G is what we teach our students. That's from the word gyr, because there's a lot of speckled animals in that gyr breed. And that the last slide that Joel showed you, those are a beautiful set of gyr cattle. And yeah, they're very common to have speckling happen in those. Then the Simmental pattern, you've probably seen that too. It's similar to Hereford, right? But more of a blaze face rather than a full white face. Certainly less white. What I've noticed on those two breeds is their colorings have sort of come together. Other than Simmental allows the registration of black animals now too, right? There's a separate breed association for black Hereford now. If you don't believe me, go Google it and see it, you know. Black Hereford, in fact, I've got students, and my student in North Dakota State is doing EPDs for that group, okay? And at this point, you know, we're just in early stages of that. Finally, the spotting you see in Holstein is recessive, okay? So it could hide when you pair it with the solid gene, okay? So, but that, if they have two copies of that small recessive S, that you get the paint all over, kind of like Holstein, what you see. Another one, you know, we mentioned Murray Gray a while ago. Murray Gray is known to be sort of descendants of Angus and Shorthorn, kind of bred together for many years. And so Shorthorn is very common to have roning. And let's use this, these genotypes that kind of give you alleles of big R and little r. And by the way, the first one is completely wrong. Little r would be solid, okay? The recessive condition there. But you can think of each big R as adding some white to the mix. So big R, little r, the heterozygote would result in roan, and roan is like white hairs interspersed in coat of other color, okay? So, and I know that there was this Brahmin Shorthorn project before the Brahmin Charlet project of Dr. Peacock. So there was a Brahmin Shorthorn project that Marvin Koger led here beginning in the 60s at ONA. So I'm suspecting also that there's some roning contributing to your white Angus here. And seeing the Murray Gray, and that just reminded me, the Murray Gray sort of remind me of some of your cattle, you know? So I think they have a little more Shorthorn influence in them, though, obviously. And big R is white. Oftentimes mutations that cause white in livestock also affect fertility. Have you heard that before? Particularly in horses. Like a dominant white horse, there's some fertility issues associated with that color. So color inheritance in cattle is very simple, right? It's a big phenotype, we can measure it, but it's not as simple as we thought originally. Probably more quantitative in nature than we ever believed possible before. And just throw this out at you, you've probably seen similar animals. This one is a white brindle, okay? So how does that happen? If brindle is black hair is patterned into non-black coat color, then how does white occur? And I don't know the answer to that, you know, but I see it. I've seen it a lot. In this animal as well. You know, you get some white brindling. How about that? How do I explain that? You know, it looks very Hereford pattern to me, right? Except red, you know, when you, in straight Brahman, they're not necessarily red. I mean, there's some that are sort of red tinged, you know. I know several, I've got friends in Florida who breed cattle, Brahman cattle that often have that red tinge to them, you know. They also tend to be ones with black tipped ears, okay. Is that what this is? I have no idea. But it looks almost like there's some spotting happening there. So what we really thought was not complex, I think is more complex than we ever knew before. So this is one of our Nellore crosses, and I want to thank you for paying attention to me. If you weren't paying attention, you were doing a good job of an impression of someone paying attention, and I really appreciate it. But if you have questions, I would be happy for you to email me, and I would do my very best to answer them if I could.